everybody, today we are debating creationist genetics, and we are starting right now. Ladies and gentlemen, thrilled to have you here for another epic debate where at Modern Day Debate we host debates on science, politics, and religion. And this one should be a very interesting one. In particular, this is a backlash debate. What we mean by that is that Dr. Dan and Sal have engaged on similar topics within this ballpark before. That's linked in the description if you love this topic and want to hear that in case you haven't already. And so this is kind of a continuing battle on these topics, and so it should be a lot of fun. Want to let you know, though, if it's your first time here, consider hitting that subscribe button as we have many more debates coming up. We are very excited about, for example, tomorrow it will be, is the God of the Hebrew people immoral or evil? That should be an interesting one. And then Friday we will be back for a feminism debate, feminism on trial, as we have two female speakers that will be hashing it out. That should be a lot of fun. And also want to let you know, if you happen to have any questions today, feel free to fire it into the old live chat. And if you take it with at Modern Day Debate, it makes it easier for me to get every single question into that list for the Q&A. Super chat is also an option, in which case you can make not only a question but a comment for the Q&A, and it'll also push your question or comment to the top of the list. And with that, want to let you know, we are very excited. I will, in just a moment, so what I'll do is hand it over to Erika for the actual moderating. I'll be keeping an eye on the live chat and also kind of collecting questions for her and I to alternate asking the speakers during the Q&A. But I am excited to introduce Erika, and then she will be giving Dr. Dan and Sal a chance to introduce themselves. I want to first say thanks so much to our speakers and our co-mod. All three of you, thank you very much, Dr. Dan, Sal, and Erika, just for being here tonight. It's going to be a great time.
Man, nothing is more fun than coming on Modern Day Debate and like nerding out about science stuff. I can't think of a place I would rather be with these two based and of course epic speakers to chat about genetics. I'm very psyched, and I hope everyone out there is psyched as well. Moderation, I've moderated before, but not to the degree that we'll be doing today. So I'm excited and I'm ready to go. So Sal is going to introduce himself first. So Sal.
One second. Forgive me for just one thing that I would just have to say we are so excited about this, Erika. I know that you maybe don't like to be in the spotlight, but we are so excited for you, Erika. I don't think you've been on since the last time that you've been on. This unique and special thing happened in your life. We're very excited. So I don't know if you guys have heard. So I have to say this, folks. We are so excited. We are celebrating with you. So, I mean, Erika, I'll let you say it yourself because it's your special thing. We're so excited for you.
James has put me on the spot, but I am very excited and happy to announce to the MDD fam, as the youngsters say these days, that I got engaged. So there is indeed someone out there who in real life actually likes just like listening to me talk about like monkeys and stuff like that. So it's great. I'm very happy. One of these days I'll drag him into the frame so everyone will have to feast their eyes on the person that is on the guy that's gonna be my spouse. So.
Well, I've got good news, Erika. We may actually, a lot of people don't know this, we have at Modern Day Debate, we have our own journalists, and it turns out that they were actually on your trip, and they happened to see this special event. And so we, as a way of celebrating with you, Erika, we have to show the audience this is really important. So folks, you may be surprised. I have to show, everybody has to see this. So give me one moment as we switch over. So this is apparently what our journalists had found. This is the extremely exciting engagement event. It's a small world, I guess. We were surprised. Is this legal? But this is definitely surprising.
James.
But
oh my God.
You have to say, so congrats to both of you. It's
oh my God.
It's 2020. I mean, anything.
I can't believe you guys found out. I mean, did you guys find out that the honeymoon was planned for Dinosaur Adventureland? Is that how this came to light?
That's
Did you just find that out?
that's exactly what it was. We just never knew. It's crazy, Erika, but we're happy for you. We are always there for
Oh
you.
my
This is
gosh.
special.
Ugh.
Is it okay if I show the real picture? I mean, okay, well, I guess you feel
That's
pressured to say yes. Sorry
yes,
about that.
no,
But
no, I think that would be fine. Yeah, you can let's like light dox him. Show
I lightly, yes. I folks, this isn't the real picture. Let me show you the real picture. It's no more jokes. This one, I will show you the actual picture. Because it's such a great picture. So let me just pull it up. This is the real picture. So, and well, Erika, trust me that it's the real picture. I hadn't pulled it into Zoom, but we're so excited for you, Erika. So very exciting.
Thank you so much.
And really special. I'm sure he's a terrific guy, so we're just really excited for you. And so with that,
Oh my god. Ugh, I'm probably as red as my shirt. Good god.
we will be kicking it over now to Erika, our moderator, as she will be, as I had said, she will take the reins now. And so thanks so much, Erika, and the floor is all yours.
Oh, well, I'll do my best, but I think I just had a pulmonary embolism just then. So I'll work through it and do my best to moderate. All right, let's get this show on the road. I'm ready to talk some science, and I'm beat red as it is. So Sal, go ahead to introduce yourselves to everyone. Everyone is let us know who you are. Give us a refresher.
Oh, thank you, James, Erika, Dan. Thanks to our viewers. I hope we can put some of you into nerd heaven tonight in this debate. I'm Salvador Cordova, and I'll be representing the Christian creationist perspective. I'm a former scientist and engineer in the aerospace and defense industry, but presently do research and news reporting in the area of molecular biophysics. I work directly for world-famous applied geneticist and retired Cornell research professor John C. Sanford, whose work I'll be discussing tonight. I also do computational and research work remotely for various labs and professors around the country. Some of my work focuses on the beta-lactamase nyelinase family of enzymes and the post-translational modifications of the topoisomerase family of enzymes in cancer research. I have 5 science degrees, including an MS in physics from Johns Hopkins University and an MS equivalent in biology from both my professional work in addition to classes I've taken at the FAES Graduate School at the National Institutes of Health, where I had the privilege to study under the staff of top evolutionary biologists and bioinformaticians. I used to believe in evolution. I no longer do. I self-identify as a Christian creationist.
Okay, awesome. All right, Dan, your turn.
Hi, everybody. Thank you for watching, and especially thank you for the returning audience. Thank you for coming back for round 2. And thank you again, James, for hosting, and Sal and Erika for joining us here. So my name is Dan Stern-Cardinal. I'm an evolutionary biologist. I have a PhD in molecular genetics and microbiology, and my graduate research was on viral evolution, mutation accumulation, and fitness effects. So this topic, genetic entropy, is right in my wheelhouse with what I like to talk about. So I'm thrilled to get going.
All right. So with that, I think we're going to launch into our openings. Now, Sal's going to start off for us. We've got 10-minute openers, and I'm gonna crack down on that. Don't go 1 second over 10 minutes, or you know how I am. So all right, Sal, just whenever you start, I'll start the timer, and I know you've got a timer on that end too. So we'll just hold each other accountable.
This is part 2 of a debate about genetic entropy and creationist genetics. Some follow-up references to this debate are available at the website www.evidenceandreasons.org. Even though the website is still being built out, the website does have further material about the issues laid out in this debate, and more material will be added periodically. To recap a bit, the primary focus of tonight's debate is about the hypothesis of genetic entropy, and secondarily about creationism. The genetic entropy hypothesis was put forward by retired Cornell research professor John Sanford, who is also presently my employer. The primary claim of Dr. Sanford's book is that the human genome is slowly deteriorating with each generation. Dr. Sanford was a Cornell research professor during much of his 45-year career as an applied geneticist. A sizable fraction of all genetically engineered organisms were created through his inventions, which helped feed starving billions. His inventions are in the collection of the Smithsonian National Museum of American History. Dr. Sanford was an atheist, but at age 39 became a Christian, and by age 49 had become a Christian creationist. His knowledge of applied genetics contributed to his present belief in creationism and his rejection of evolutionary theory. Professor Sanford is among a growing number of scholars who are creationists, but who attain prestigious scientific credentials. For example, Professor of Molecular Biology Chang Tan is an Ivy League PhD and postdoc from Harvard, and I'm going to be working with her, so I'm excited about that. Rob Stadler is an accomplished biomedical engineer and scientist whose medical devices have been implanted in millions of patients. Pictured here are a couple of first-rate books by the individuals just mentioned. In 2017, Emeritus Professor of Mathematics Bill Basner, along with John Sanford, published in the Journal of Mathematical Biology a corrected version of Fisher's fundamental theorem of natural selection. The theorem they proved is stated right there at the bottom. This is a fragment of the proof of that theorem. Their article on the theorem set a record for the number of downloads of any article in the Journal of Mathematical Biology's history. This is all the more amazing given that it was these two creationists who set this record by publishing a mathematical correction to a long-standing theorem in evolutionary biology. This theorem is part of a larger body of literature by Dr. Sanford and colleagues that has advanced the genetic entropy hypothesis and other criticisms of evolutionary biology in peer-reviewed scientific literature. Their article resulted in Bill Basner becoming keynote speaker at this International Biology Conference in 2018, and also later that same year, Dr. Sanford getting invited to speak at the National Institutes of Health. But independent of Dr. Sanford's work, consider some of the statements by top evolutionary biologists about genetic deterioration in humans. Michael Lynch, probably one of the most respected evolutionary biologists, had this to say: "It remains difficult to escape the conclusion that numerous physical and psychological attributes are likely to slowly deteriorate in technologically advanced societies. The incidences of a variety of afflictions, including autism, male infertility, asthma, immune system disorders, diabetes, etc., already exhibit increases exceeding the expected rate. This observational work may substantially underestimate the mutational vulnerability of the world's most complex organ, the human brain. Because human brain function is governed by the expression of thousands of genes, the germline mutation rate to psychological disorders may be unusually high. At least 30% of individuals with autism spectrum disorders appear to acquire such behaviors by de novo mutation. It has been suggested that there has been a slow decline in intelligence in the United States and the United Kingdom over the past century." My translation of this is, we're getting sicker and dumber. So how can Darwinian evolution, how can Darwinism fail to arrest this problem? I'm going to illustrate it. Suppose 20% of a population acquires new mutations, slight birth defects in each generation. Now let's suppose we have perfect selection and it eliminates 20% of the population that are mutants. And I'll illustrate that by invoking the Terminator. And he's going to say, "Hasta la vista, baby." And he's going to eliminate one of the mutants. And "Sayonara, muchacho." And he's going to eliminate the other mutant. And what remains are the survivors are free of mutations, and they are become parents of the next generation. So that's one scenario. What if we have a scenario where all the offspring have new mutations? They have some slight birth defect that their parents didn't have, a defect that may not be noticeable outwardly, but it is a slight defect in their genome. So what will happen in this scenario? Well, we certainly can't, perfect selection would wipe out everyone, and that wouldn't be good either. So what is the recourse? The recourse is we have imperfect selection, where we just take out maybe the worst, but the remaining still have some slight defects. The species survives, but each generation is dumber and sicker. So this is a problem. This is not a creationist theory. This is something that is recognized in evolutionary biology, as illustrated by this observation by Dan Grauer in 2016. He said, the genome of each human newborn carries 56 to 103 point mutations that are not found in either of the two parental genomes. If 80% of the genome is functional as trumpeted by ENCODE Project Consortium 2012, then 45 to 82 deleterious mutations arise per generation. And he observed the consequence of this in his talk in 2013. 3 problems. If the human genome is indeed devoid of junk DNA, as implied by the ENCODE project, then a long, undirected evolutionary process cannot explain the human genome. If, on the other hand, organisms are designed, then all DNA, or as much as possible, is expected to exhibit function. If ENCODE is right, then evolution is wrong. And we'll talk a little bit more in this debate about the ENCODE Project Consortium. It was so far a $500 million project by the National Institutes of Health. So let me illustrate the problem again. Assuming we have, let's say, I'll just pick the number since he mentioned 82 mutations. Let's suppose it's 80 mutations. There's some parameters here we could talk about that shows why this little simplification is valid. But suppose at generation 0, we have this parent free of birth defects, and that parent has a child with 60 mutations and has even more children. But on average, all the children have about 80 mutations. So even if we had perfect selection and wiped out, well, the parent passes away and perfect selection comes along and wipes out the worst, but even the best of the worst of the defective children survives and has mutations and becomes a parent to the next generation. So we will repeat this process, and it should be evident that, as Dan Grauer said, if we have a mutation rate of 80 per individual per generation, there'll be genetic deterioration. And all I'm doing is illustrating this. So parent dies and we have elimination of the worst, but still, even under survival of the fittest, the genome keeps deteriorating. And I'm just going to keep repeating this, and you can see the numbers increasing. This is something Dan Grauer observed. I know I have a minute and a half and I'm almost done. So fast forward to million generations, we'll have 60 million mutations at that point. And this one solution to this problem was proposed by Susumu Ono. This was understood by many population geneticists that we cannot tolerate too many mutations. So they said one solution to this is to invoke junk DNA. And he said there seems to be a strict upper limit for the number of gene loci which we can afford to keep in our genome. Consequently, only a fraction of our DNA appears to function as genes. And I'm going to skip a little bit. It then follows that the moment we acquire 10 to the 5th gene loci, the overall deleterious mutation rate per genome becomes 1.0, which appears to represent an unbearably heavy genetic load. And that's an archaic version of what Dan Grauer was saying. Dan Grauer had it more up to date. And I'll close my opening with this. Why was the junk DNA hypothesis postulated? And my answer is the idea of large amounts of functional DNA is incompatible with evolutionary theory. And I think that sets the lines of the debate for this evening. And I'll yield the floor to my distinguished colleague, Professor Dan Stern-Cardinal. Thank you.
All right. Okay, thank you, Sal. I really liked those little nuclear explosion animations. I'm always a big fan of animations being incorporated into things. So Dan, you're muted right now, so you want to unmute yourself first so that we can hear you.
Yes, I do. Thank you.
There you go. All right, so Dan, when you start talking, I'll hit the clock.
All right, just want to check.
I'll give you 2 minutes, if that's all right.
Just want to check. You should be able to see the PowerPoint slide, yes?
Yes, we can see it.
Wonderful. All right, here we go. So thank you, everybody, for coming back for round 2. I'm going to start with a little bit of background information, but I want to recommend that if you haven't watched round 1, Go back and watch the intros, our opening arguments from round one. They were longer and they're going to be a little bit more detailed. So just strongly recommend you do that before crashing right into this, because we're kind of both building off of round one. So we're talking about genetic entropy here. As Sal said, this was the idea proposed by Dr. John Sanford in his 2005 book. And the idea here is that within a population, mutations accumulate over generations. As a result, fitness declines, and this will ultimately lead to extinction. Now, the relevant topic here, the relevant underlying idea is evolutionary fitness. And evolutionary fitness is reproductive success. Basically, how many viable offspring do you have? Offspring who they themselves will reproduce. The central claim of genetic entropy is that mutations cause reproductive output to inevitably decline over generations, and this will ultimately lead to extinction. Now, there are other ways to talk about fitness besides an evolutionary context. You could talk about things like function or structure of different things. You could talk about that kind of at a macro scale or at a genetic scale. Talk about information in the genome or something like health or intelligence. But all of those things are upstream of reproduction. At the end of the day, the question is long-term viability. Is a population going to be able to survive, or is it going to go extinct? That's the question that Sanford tries to address, and his answer is inevitable extinction. The idea with his argument is that he is trying to refute evolutionary theory as a broader idea. But if you have to redefine fitness into something different in order to do that, then that shows how weak the argument is. You can't refute an idea by redefining the terms into something that suits your liking better. You have to engage with the idea as it exists. So from part one, Sal's argument, which we heard right here, is that fitness, it shouldn't be about reproduction. It should be about something else. Health of the organism, engineered features of the organism, intelligence, health, structural precision at the cellular or genetic level. And defined that way, most mutations are harmful and the genome is too constrained. And this is what Sal was just saying, the genome is too constrained to be able to tolerate very many mutations. Therefore, the genome is degenerating over generations, and that is the concept of genetic entropy. So there are a couple of problems with this idea. First is that that is not John Sanford's argument in genetic entropy. He talks very specifically about mutations and the genome itself, not about other aspects of an organism's well-being. That also changing the terms does not address the problems with Sanford's argument. The shortcomings that I'm going to discuss in a minute apply equally to Sal's version of the argument. So here are the specific components of Sanford's hypothesis. First is that virtually all mutations are harmful. These fitness effects are constant. The accumulation of these mutations is approximately linear. The rate of accumulation and the relative frequency of harmful mutations does not change generation over generation. And this accumulation over generations is inevitable. Selection can't slow the accumulation nor remove mutations once they have occurred. I raised a number of specific objections to these ideas last time that were not directly addressed, so I want to reiterate them briefly here in the hopes that we can address them in round two. So the first objection is that fitness effects are not fixed and beneficial mutations unquestionably exist. So for example, in humans, lactase persistence, that is a beneficial mutation. There are no downsides to that mutation. Contrary to what creationists claim, there are no loss of regulatory mechanisms or anything associated with that mutation. That's just straight up beneficial. But there are also mutations whose effects are context specific. So the mutation that causes sickle cell disease is very harmful in people who are homozygous for that trait, who have two of the mutations. But if you're heterozygous, if you only have one, you don't have the disease, but you're resistant to malaria. So if you live in a malaria endemic area, that trait will actually be selected for being a heterozygote. In influenza, we can look at mutations that confer resistance to Tamiflu, and there can be a cost to those, but you can have secondary mutations that recover the fitness cost. And then in elephants, I love this one, in elephants, the tuskless trait is very bad because they use their tusks to do things like root around for food. But in a context where humans are constantly hunting elephants for their tusks, being tuskless is actually really helpful. So context matters and beneficial mutations exist. The second problem is that the fitness effects reach equilibrium. Sanford claims that over time, as mutations accumulate, the fraction of deleterious mutations stays at approximately 100%. But that's just mathematically not possible. As a harmful mutation occurs, it's replaced by the potential reverse mutation in the universe of potential future mutations. So over time, as harmful mutations accumulate, you approach an equilibrium where the frequency of helpful and harmful is approximately equal. And that's not bio, that's not anything specific to genetics, that's just basic math. The third problem is that selection occurs. According to Sanford, everybody deteriorates approximately equally enough that selection can't distinguish between them. So mutations accumulate even when you hit a threshold of viability. Even after that point, mutations continue to accumulate. But in reality, populations have variation. So as some individuals cross that threshold of viability, other individuals stay just below it, and you have selection against the inviable genotypes. So what you end up with is a population that hovers right on that knife's edge of viability, and that's called mutation selection balance. So for those reasons, Sanford's reasoning is completely wrong. So my goals for part 2 of this debate are: 1, provide Sal an opportunity to respond to those specific shortcomings in Sanford's argument. 2nd, establish the correct definition for fitness and show that Sal's new definition doesn't solve the problems created for the creationist argument that the correct definition causes. 3rd, examine the supposed limits of evolution, things like a viable mutation load, the percentage of the genome that could potentially be functional, and if we have time for it, things like generating and maintaining information or traits in the face of constant mutations. And then finally, if we have time, I'd love to talk about specific supposed examples of genetic entropy, but we'll see how the discussion goes and see if we get there. So I want to end this opening argument with some specific questions for Sal that I hope we can answer as we go forward. 1, do you disagree with any of the 3 objections I raised last time and just reiterated here? 2, on fitness, do you disagree that the outcome of genetic entropy ultimately is genomic deterioration and eventually extinction? 3, what, if any, are the limits and thresholds of evolutionary processes in terms of like mutation load and percent of the genome that could be functional and still be viable over the long term? And in particular, what are the evidence for these limits? And then finally, can you identify any specific instances of genetic entropy? So a couple of related topics that I hope we get to. You mentioned the 2018 Basner and Sanford paper on Fisher. I would love to talk about that. I don't know if we'll get to the idea of information in genetics and biology, but we'll see if we get there. Epistasis may come up. ENCODE, I'm sure we're going to talk about ENCODE, although I really think that's kind of a distraction because, as we'll see, I don't think there's good evidence that there is a limit to viability dependent upon what fraction of the genome is functional. And then finally, we could possibly, if we get there, talk about environmental change, the idea that, well, it's not that organisms are just getting bad on their own, it's that they're getting kind of specific to their environment. They're becoming specialists rather than gradualists, and then if things change, things go downhill. That has its own problems for genetic entropy because that's not the argument that Sanford makes. So we can get to that if we have time. And with that, I'll turn the floor back over and it's back to Sal. Thank you.
One thing I just want to quick jump in and mention, because I forgot to mention it at the start, I have put all of the links of the speakers in the description, folks. So that way, if you're listening and you're like, I like that, you can hear plenty more, including Erika. Her link is down there as well. So all 3 of our guests today are in the description. Want to let you know that. Also, one really quick thing, just as I have to mention, I've been moving lately, so I'm like super behind on email. So I seriously hope nobody takes offense to that. I try to answer every email unless it's attacking me ferociously. I usually ignore those, but I try to answer everyone. And so sorry if anybody's waiting on messages. I really do try to get back to everybody. And so with that, thanks so much. And back to Erika, who will keep us rolling along.
All right, that was exactly 9 minutes by my count. So I was getting ready to say 1 minute warning at 9 minutes, and then you finished up, and it was perfect. I didn't have to say anything to either of you. You guys nailed it. Okay, so I believe we're going to start with Sal. And for those of you out there in the ether wondering kind of how things are going to go today, both of our speakers have kind of discussed slightly what they want to talk about, broadly speaking and topic wise. So we're going to kind of let them take the lead on what we're speaking about, and they'll have about 20-ish minutes per topic, give or take. So that we make sure we don't stay on a topic for too long, which, these guys are pros. They know what they're doing. They were like, these are the topics we're discussing, and we're going to get through all of them, dang it. So I'm going to toss it over to Sal and let him kick us off.
Dan, is it okay? I'm going to give a presentation for about 10 minutes, and then we can fill out. Okay, great.
Yeah, go for
That's
it.
great.
Yeah, I'll start a timer kind of in the background and let you guys know when we're like at 15 minutes, and then when we're at 20, if that's cool with you.
That's cool. Hopefully you can see my slide.
Yeah, we got it.
Is junk DNA really junk? And this was a slide from by Professor Dan from our first debate, and he gives a listing of things, his idea of how much is functional in some sense, and certainly in what's not. And I think that really defines the debate tonight, and that's what I'm going to have to. The next 10 minutes is going to be nerd heaven for some of you, because this is just going to be pure technical stuff. So as I mentioned, as I alluded to, the junk DNA hypothesis came out. This is kind of an archaic form. There's a more modern form. And I read that by Dan Grauer. It regards functionality. So I'm going to talk about functionality tonight, and I'm going to try to do just in this first 10 minutes is just to do a setup. I'm not going to actually respond to all of this here, because that's going to take 20 minutes. So I'm going to break this up into two 10-minute segments. So if ENCODE is right, then evolution is wrong. And that really, if the supposed junk DNA is mostly functional, then evolution is wrong. I'm going to actually argue, in addition to that, evolution is wrong, even if only 11% is functional, and that is the figure that Professor Dan provided. And the reason for that is this table that can be derived from population genetics. It uses the same math that Professor Grauer used to criticize ENCODE. So Professor Grauer said if ENCODE's right, every female has to give birth to 10 to the 35th children. And another geneticist said, well, maybe the mutation rate's only 3 per kid, but the females still need to have 40.2 kids. If we use Professor Dan's figures from the debate on this past June, that would still be 119,748 children per female. The math works out, by the way, that the percent functionality is about equal to the number of harmful mutations per individual per generation. I should highlight that even slight changes in percentage, even 1 or 2, 3, 4%, whatever, would be tremendous increases in the number of children that each female has to bear. So even a 3% increase from the figure of 11 mutations or 11% per individual per generation would result in an increase of 20-fold. So again, this is really, I'm trying to lay out what the lines of disagreement are. So intuitively, why did people suspect that, even though Ono said that most of our genome's junk, why were there suspicions that came about? It wasn't a creationist conspiracy. From a 2009 paper by director of the NIH, Francis Collins, his research resulted in something I'm going to quote from another paper, but it resulted that the explanation, the consequence of this is what is emerging from these GWAS. GWAS is genome-wide association studies. However, is that 90% of disease-associated SNPs, that's single nucleotide polymorphisms, we can call them mutations, that's close enough, are located in non-coding regions of the genome. So that's not absolute proof that the non-coding or junk regions are functional, but it gave rise to a lot of suspicions. So let me continue with that. By the way, I have 6 hours of lectures on these topics at this website, evidenceandreasons.org. I talk about Muller's limit, Fisher's theorem, and protein probabilities. So some of this is actually discussed in detail. This is Waddington. He coined the term epigenetics, and a lot of ENCODE deals with epigenetics. The idea is that DNA is not just a blueprint for proteins. DNA is a scaffold for machines. So here's some pictures of scaffolds. DNA is a scaffold for machines. DNA is also designated parking spaces and addresses with names and motifs for machines. And this is the topic of the ENCODE 4D nucleome project. DNA is also part of a DNA chromatin computer. So DNA is part of a computational complex. And I mentioned chromatin. This is a conceptual diagram of chromatin. Chromatin is made of DNA here. And in eukaryotic cells, the DNA winds around things called histones. And these are histones. And the histone with a DNA is what we call a nucleosome. And nucleosomes make chromatin. There are modifications on the DNA that can happen. These methyl modifications on the DNA. And I'll picture that here. And we can call these one form of epigenetic modification. It's a chemical modification on the DNA. And this is an illustration of what will happen to these methyl modifications in a mouse embryo. So 3 hours after fertilization, these lit up DNA blobs here. This is the paternal DNA from the daddy mouse and the mommy mouse here. After 6 hours, it's lit up a little more, but then after 8 hours, the methylation marks go away on the paternal DNA. So this is some sort of DNA computation going on. You can actually visualize it. So this is a great experiment. We're able to visualize the changes in this chromatin computer. So there's a little reboot here. That's kind of cute. So why am I pointing this out? This DNA then, even in non-coding regions, can be used as part of a computational complex. And that'll lead into why a lot of this junk DNA isn't junk. So again, I'm going to focus on these nucleosomes and these histones. These histones have little tails. These tails are actually memory tails. They can be written to with bits, and these are all the ways that it can be written to. That's the little histone tail. It can also be read. So there's readers and writers on this memory of the histone. And by the way, see that wire there, that blue wire, that's DNA. So DNA is part of this computational complex of readers, writers, and erasers. And that's not a creationist term, that's the term in biological literature for what's going on in this DNA chromatin computer. And in the Stem Cells Handbook, they actually use the word, read-only memory and random access memory. So the traditional view of DNA is the read-only memory that codes for proteins, that makes proteins. It's the blueprint for proteins. But then this epigenetic layer of the modifications I showed, they use the word epigenetic memory. So that's the chromatin computation. I'm going to show, one thing I'll say is we call this chromatin structure here because it looks like beads on a string. And I'm going to show a little picture of that with an electron micrograph. We have these nucleosome core particles. This is the chromatin. The wiry type stuff is the DNA that connects it. I call it God-made random access memory. And it's just kind of cute. It resembles man-made random access memory way back in the 70s for magnetic core memory, and even the abacus in ancient times. So there's, again, I'm saying this is a chromatin computation. I have 2 minutes left. These are some of the chemical details. Last time I talked about the hot air modification. So this is a karyotype of a human. From chromosome 12 emerges a link RNA. The link RNA goes to it travels to chromosome 2 on the winds of Brownian motion. It parks there. Like I said, it parks. DNA can act like a parking lot. When it's there at the Hox cluster of chromosome 2, it recruits this polycomb repression complex and it modifies the histone. When that histone is modified through this computation, it changes. When it's done in one case, it will change the quality of the skin. So that's why the quality of the skin is different between the eyelids and the feet. So that's a little bit about the chromatin computation. And I have about Dan, if I could have 2 minutes, and I'm just going to cut off. I'm going to go over a little bit. We could talk about this. Hopefully this is not controversial. We have stem cells that come from the embryo, the totipotent stem cells differentiate into all these kinds of these other cells, cell types that make up our various tissues. They go in organs. And there's another picture of that. I'm going to show the chromatin computation here. This is a beautiful picture of it. Pictured here is the embryo of a nematode worm, a worm of some sort. And the little thing glowing here is a cell that is going to be the parent to a cell lineage of the germline cells, which are basically reproductive cells. And I'm just going to zoom in a little bit. You could see that as the color changes, we're having a different, the state of the chromatin computer is changing. And so at some point, you can actually see how it's gone from green to red. These cells now are in a different state. They're going to end up being the gametes that go in the gonads. So that's a little bit about the chromatin computation. And that's all I had to say, because I don't want to take up this segment, but that is a little taste of where I'm going. It's going to be in another segment that I'm going to discuss. This forms the basis why I don't think there's as much junk as the evolutionary biologists claim. So, Dan, thank you.
Thank you. All right.
Roger that.
There we go.
Thanks so much, Sal. It's helpful that we've got the images, I think. It allows us to really appreciate what you guys are saying, particularly when it goes into some of the more less layman genetics. I don't know if I can speak for everyone, but I definitely appreciate it. So, all right, Dan, when you start talking,
right. I got a clock running. So bunch of things in response to that. First, I agree that cells are complicated, but complexity is not itself an argument against evolution. Evolution doesn't have a problem with complexity. So in order to make an anti-evolution argument, one needs to go the next step and show that it's actually incompatible in some way. And there's a few stabs at that here, but none of them work. So first thing, I just want to correct a couple small things. So in the slide of mine that you showed, it didn't say it explicitly on there, but or maybe it did, I don't remember. But it had about 11% of the genome being functional, and you said that works out to about 11 harmful mutations per generation if you get about 100 per generation. The faulty assumption there is that every mutation in a functional region is going to be harmful in some way. And I think we should be able to agree that that is simply not true. So I provided the example of lactase persistence, the ability to digest lactose throughout one's life. That's obviously a beneficial mutation. Another example is that there are two alleles in Tibetan populations that facilitate living at very high altitude. They improve oxygen saturation in the blood. These are just like very obviously beneficial mutations. And then there's just tons of variation that has no physiological effects. There's just lots of variants. And I'm just going to ask you directly, Sal, I mean, can we agree that there are mutations in functional parts of the genome that are either beneficial or neutral? Like, can we agree on that? I'm not looking for like a super long answer.
That's
like,
a very
try
good
to
question,
be quick.
and it deserves an answer. I think the best way to frame it is, as you said, that there'll be mutations where there is no immediate physiological effect.
Well, is there, I'm not going to let you off the hook on that, though. Is there a long-term physiological harm to lactase persistence? The answer is no, there just isn't. Is there long-term physiological harm to being able to breathe efficiently at a higher altitude? No, we know what those alleles are. We know what they do. There's no harm to it. There are alleles that are documented that just don't have a cost. So that's the first thing, is that we need to establish that just because a mutation occurs in a functional region, that doesn't mean that it is harmful. We know of beneficial and neutral mutations in those regions. The second thing, and I'm going to pull up a slide here for this, is that disease associated doesn't imply functional. So I'm just going to pull up one of the slides here. And this is a paper from just earlier in 2020 looking at the relationship between imprinting, which you mentioned, and which involves methylation, and an immune response to retroviruses that invade our genome. Now, a lot of the activity that ENCODE documented was around things like methylation. And a lot of this actually takes place at ERVs, endogenous retroviruses. So like right here, that's an ERV. Well, that activity doesn't imply that that thing is functional in the context of human physiology, because the problems that could arise from that are that when those proteins are expressed, those are viral proteins. So if those get expressed, they could actually hurt you, because that's the virus trying to hijack your cells to replicate itself. So being a target for something like that, and when it mutates and can no longer be a target for that because its sequence has changed, so our proteins are not able to recognize it as a target, when that happens and it causes disease, that's not an indication on its own that that region is functional. And in the case of transposons and endogenous retroviruses, which collectively are over half of our genome, that reality being disease associated, which is not in many cases, but it is in some of them, that's an argument against their being functional, not for it, because these are genomic parasites. So if we can shut them down, they can't do what they want to do. I'm personifying them, want. But if they mutate and we can't shut them down, there could be a disease phenotype associated with that. So that's why that's not a good argument for functionality. The other thing I want to get into, though, the main thing I want to respond to here is the idea that we can only tolerate so much in terms of the functional fraction of our genome. So I want to just give me a second here. I'll find it. Here we go. This is the one. If ENCODE is right, evolution is wrong. I think Dan Grauer was completely wrong when he said that. And we have evidence of this now from a paper from just this past April, Mutational Load and Functional Fraction of the Human Genome. And what the authors did in this paper was a very cool model. What they did is we examined whether and how an analysis based on mutational load might set a limit on functional fraction. So we now have people that are directly investigating this question of, is there an upper limit to what we can tolerate given mutational load? They said, we find that the functional fraction is not very likely to be limited substantially by mutational load, and that any such limit, if it exists, depends strongly on the selection coefficients of new deleterious mutations. Now, is this relevant to genetic entropy? It absolutely is, because this actually works the way Sanford says and the way you've described in terms of every genome getting mutations every generation. So what they said is that our approach is different because they compared mean fitness at mutation selection equilibrium with the fitness of an individual, not with the fitness of an individual who has no deleterious mutations. We show that such an individual is exceedingly unlikely to exist. So they have everybody experiencing deleterious mutations in this model. They just have it different for each individual. In other words, what I described earlier, there's variation, and therefore there's differences in fitness. The cool thing about this model is it directly shows what we've seen experimentally. So last time I described this paper, Springman et al., 2010, this was a paper where the researchers mutagenized a population of viruses. And what they showed is that you'd expect the saturated with mutations population to go extinct. Instead, most of it lost fitness, but a few rare individuals ended up being very high fitness. And the model in this paper mirrored those exact results, where you end up having fitness peaks that are very high, but they're very rare. It's a low frequency genotype. So how does this work? This seems so counterintuitive. It works because as you increase the functional fraction, you get stronger selection. So when you find those rare beneficial mutations, even in the context of a background that has harmful mutations, you're better able to take advantage of that and select for them. So this is a table from the paper that just shows that S is the strength of selection. It's the selection differential for anyone watching. It's just how strong is selection. And then this is the functional fraction, anywhere from 2% of the genome up to 100% of the genome. And the bigger this number is, the higher the maximum fitness of that population, showing that as you make the genome more selectable, you can actually do a better job finding those high fitness genotypes. Also, the cool thing about this is it doesn't matter what your definition of fitness is. It can be structural, it can be about the 4D nucleome, it doesn't matter. It's just functional related to constraints on the DNA sequence. That's all that matters. So the authors summarize, calculations that purport to establish a load should, in our view, be based on the distribution of actual fitnesses that are expected in a real population. As we have shown, the properties of this distribution depend not just on the number of de novo deleterious mutations, but also on the selection coefficient against deleterious mutations. In other words, selection exists even if everybody in the population is experiencing harmful mutations. And this is exactly that third thing that I brought up as a problem with Dr. Sanford's model, because Sanford considers the mutations and how they accumulate, but he ignores the selection. And that's the problem that this paper addresses. And that gets around the problem of, well, if the genome is X% functional, it's not viable over generations. That's just simply not true. And this is a model that shows it, and it comports with empirical data that we've generated experimentally.
Okay, all right. So Sal, would you like to take your next 10 minutes and respond to that, or would you rather have an open dialogue?
I think we agreed that Dan can now put something on the table. Is that right, Dan? Did you want to put
just so I will go to my next thing. It's not so much like a point as just I want to just like settle questions. So what I really want to talk about here are the questions I felt like were left over from our last round. So here, I'm just going to put these questions up and we can go through them. And this isn't going to be like a 10-minute thing. This is just going to be back and forth because I want to see what you think of these questions.
So the way I think this should go is Dan, you'll take the lead for the next 20 minutes and you get to,
yeah.
Okay.
All yeah. Right. So the way I'm going to take this section is I want to address these specific shortcomings with Sanford's model, and I want to see what you think of the underlying questions here, because I honestly don't know for some of these questions what your answer is to them, and I want to establish those answers. So basically, here are the prior questions. Are fitness effects constant? Are there virtually zero selectable beneficial mutations? Does the frequency of harmful mutations remain constant even as they accumulate? And do populations experiencing random mutations have uniform fitness across everybody? There are right and wrong answers to these questions. The correct answers are that no, fitness effects are not constant. There's epistasis. So if you have a mutation, it has one effect. If you have a mutation with another mutation, it has a different effect. That's what I showed earlier with the Tamiflu example. There's the heterozygote advantage. That's the sickle cell example. So it's harmful if you have two, but it's not harmful if you have one. And then there's environmental constraints, things like what predator is present, for example. Are there virtually zero selectable beneficial mutations? No, we should be able to agree that there are beneficial mutations that exist. At the viral level, which I really like, there's a teensy protein in HIV called VPU. And in HIV-1 group M, this protein has a brand new function. It keeps its old function. It just does that perfectly. But it acquired a completely new function to address part of the human immune system that the ancestral VPU didn't have. That's a beneficial mutation. And again, lactase persistence in the context of humans, I mean, that is a selectable beneficial mutation. It's been selected for. It's not a question of whether we can select for it. It happened. Does the frequency of harmful mutations remain constant even as they accumulate? The answer to that is no, because every time a mutation occurs, that becomes impossible. It can't occur again if it just occurred, but the reverse mutation can occur. And if the original mutation is harmful, then the reverse mutation has to be helpful, according to Sanford's own logic. So just the answer to that question is just no. And then do populations experiencing random mutations have uniform fitness? Well, mutations, I don't particularly care for the word random, but I really like the word probabilistic. Mutations are probabilistic rather than deterministic. So you don't know what mutations an individual is going to get. So when you have mutations, everyone gets hit with different mutations. That leads to variations in fitness. And even if everybody is suboptimal, you can still have selection on members of that population. So I guess my first thing is we should be able to agree on these answers. So I guess my question is, do you disagree with any of these answers? Oh, yeah, sorry. Yeah, sorry. There you go.
We want to see all your faces.
Oh, well, thank you. I disagree with the definition of fitness. I disagree with the characterization of that these beneficials can lead to some sort of equilibrium. It might be more helpful. Can we go one question at a time? I know you have it on your screen.
Yeah.
Rather than asking me, could we go point by point? That
Yeah.
I
So
could probably address that. By the way, that was a very good list. And so thank you for bringing it up.
So the first one was just, are fitness effects constant?
No.
Okay, great. So we agree on that one. Are there...
Well, in the evolutionary definition, and
Yeah.
the problems with the
Yes.
evolutionary definition that was found out by Lewontin in 2003.
I'm
when he
sure
did
we will talk about the
density-dependent
evolutionary
selection,
definition of fitness.
That's one reason he abandoned the idea of evolutionary definition of fitness, because it was a disaster. It can be characterized mathematically in any stable or sane way. He's not alone in that. Andreas Wagner. There are several other papers on this issue where the idea of evolutionary fitness is basically vacuous. And that's why I've argued for something.
I feel like we're going to be able to spend a whole 20 minutes on that.
Okay.
I think. So let's just, for here, I'm using the correct evolutionary definition. So we can agree, according to that definition, that there are variable fitness effects for a particular mutation. Okay. Yeah. Also, with that definition, are there virtually zero selectable beneficial mutations?
With that definition, no.
Okay. So far, we agree. This is not much of a debate right now. Does the frequency remain constant? So like as deleterious mutations occur, does the future frequency of deleterious mutations stay constant, or does it change as they occur?
According to that definition, no.
Okay, and then the last thing is, will a population experiencing random mutations have uniform fitness?
I'm not sure about that. What was your answer?
I mean, the correct answer is no, because not everyone gets the same mutations at once. So you're going to have variation.
I'll go with that.
Yeah. Okay.
I'm easy. Oh goodness, we're not fighting.
Okay. So we agree. We agree as long as we define fitness correctly in the evolutionary definition.
Well, I have to disagree. I think it's a terrible definition, but I'll agree as far.
Well, we're going to
evolutionary correct. I have to say that that's a little bit of a leading definition. So I just have to throw that caution but I will agree that as far as if we use the evolutionary definition, I'm not saying that it's correct. I'm not going to assent to that. But if we use that definition in that model, then we're in agreement as to the consequences. So we know why we would disagree, but within the context of it. Okay.
So we agree, as long as we're defining evolution correctly, we agree that the answer to those questions is no. And by correctly, I mean in the context of evolutionary biology.
Correctly.
Evolutionary biology has a definition of fitness, and as long as we use that definition,
I'm sorry, I'm just not going to call it correct. It's a disaster.
Well,
but I will say it's the accepted one.
It's the accepted definition in evolutionary biology that everybody uses.
You're not going to get me to say it's correct. I'll say it's the accepted definition. And I'm sorry, I'm not trying to be belligerent.
So, okay. So I want to just share one more quick slide here. That's the one. So here's the important thing with all that. Sanford's answer to all these questions is yes. He's very explicit about this in the book, Genetic Entropy. His answer to those questions is yes. And he's not playing fast and loose with the definition of fitness. He's talking about reproductive output declining over generations. And his answer to that question is yes. To all those questions is yes. And he is wrong about that.
That was in his 2008 version.
Yes, that was in his 2008 version. That's the
changed
third
a little
edition is
bit
the
in
one
his
that I'm working from.
It's changed a little bit in his 2014 version. And I'm happy to send you a copy of it.
I would love that. That would be awesome. But I just want to be clear then. So do we agree that the argument as originally articulated by Sanford in at least the first three editions, because I have the third edition, do we agree that the argument as articulated by Sanford in up to the third edition at least does not work? Do we then agree on that? And we have to we're using now a different idea of fitness to make it work.
I'd have to think about that because he did retract one thing. And so you can't ding him on that. And that had to pertain. In fact, I will, just for the reader's sake, let me see. I looked back. his 2014 edition, I'm sorry, I don't have it handy. His 2014 edition did not say all mutations are deleterious. He said almost all. So that's the 2014.
I have the quote right here. If you'll give me a second to pull it up from the 2008 edition. And remember, we're going by his argument in, whatever the genetic argument. And he says, I could even give you page from the third edition. Hang on one second. I will pull it up right here where he says that, I forget the exact wording, so I'll just not try to say it. I'll just actually pull it up. Just give me a second. Sorry, everybody watching, as we're both trying to find a reference here real fast. I just have to pull up a different set of slides. So just give me 1 second here. Play the Jeopardy music while we're waiting. Here we go.
If you're bored in the comments, go in here in the chat. Go hit.
Okay, here you go. It can be, this is page 16 of the third edition. It can be very reasonably argued that random mutations are never good. Page 16, third edition. So at least in that and the third edition, he was making the argument that
page 16, the 2014 edition, it can be very reasonably argued that random mutations are almost universally bad. So
okay, so Sanford doesn't even.
You're roboting out a little, Dan.
Dan.
Oh, sorry. I said Sanford doesn't agree with Sanford anymore. That's great. We're making progress.
Well, I wouldn't characterize it like that. I mean, everyone will change
his argument.
Verbiage.
That's fine.
I mean, he didn't agree with his own. I think that's a very uncharitable way to characterize it. But if you want to say that, that's fine. But you can't represent the 2008 as his present view.
That's fair. That's fair. He changed his view from his original argument. We agree that his original argument doesn't work.
I'm not saying it doesn't work.
Then why do you have to change it?
It's not. Well, the reason is it's not. If you have 99% of them that are deleterious, and if we're talking about the functional sense, the argument holds. So it doesn't fail on that minor correction. Just like a lot of things, with just a little bit of change in our parameters, we could still have the same conclusion. And it's actually gotten stronger over time. So you're attempting to falsify it by just saying that he changed his wording from 100% to almost always. But if 99% of your mutations are bad versus 100, so for every beneficial, if you're having, let's say, one improvement to one part of your body and the 99 other parts are going downhill, that's not good. So you can't say that his whole genetic entropy hypothesis has failed. Not to mention, I'm going to cite even more people that are in agreement with him. He's not in the minority geneticist. There's no geneticist that I know of thinks, and this is pertaining mostly to the human genome, I know of no major geneticist that thinks that in functional terms we're improving. And I gave a quote by Michael Lynch, who's very respected, and there are other people that are just right on board with Michael Lynch. So trying to characterize this as solely John Sanford's theory is, I don't think that's really fair, because it would be nice to just say, oh, this is a creationist theory, but I can name several geneticists. Michael Lynch is one. Alexei Kondrashov is another. James Crow is another. Ira Walker and Keatley have shown concerns.
That is I got to stop you there. That is a misrepresentation of their findings, and you should not cite that paper.
I'm happy to cite them.
That's not accurate.
Happy to cite them, and they may not agree on the specific details, but there's no. Can you cite it? Can you cite anyone that thinks our genome's improving?
No, but that's not the debate. The debate isn't about whether we're improving. The debate is whether we are.
Well, if we're not improving, we're either going neutral or we're going bad.
The debate is about whether we are inevitably accumulating mutations on the way to extinction. That's what.
No, we don't have to. We don't have to go to extinction to have a deterioration. You're framing it in terms of total extinction. There can be genetic deterioration without extinction. That is of concern to all of us.
Did Sanford change this line too? When selection is unable to counter the loss of information due to mutation, situation arises called error catastrophe. If not rapidly corrected, the situation leads to eventual death of the species, extinction. That's third edition, page 41. Did he change that also?
The numbering is a little bit different in this one.
I believe that's at the end of a chapter, but I don't recall specifically.
So you said page 41.
The reason this matters is actually probably leads into our next topic. So I don't want to get into, and this is for the audience wondering why we're hashing out over specific words here. The reason this matters is that the next big thing we're going to talk about, I think, is going to be the definition of fitness. And Sanford is very clear about this, that he's talking about, he states his argument in a couple of different terms depending on the chapter. So he'll talk about information, he'll talk about function, but at the end of the day, he's very clear that, and that quote is an example of it, that the end result of this is going to be extinction. And if you're talking about extinction, you're ultimately talking about reproductive output. Are you making enough offspring that are viable to replace the previous generation? So that's why this, for the audience, this may seem like a trivial point, but this matters in terms of what the actual argument is. Because if the argument is not that we're inevitably leading to extinction, then like if Sanford has changed his view on that, then like, great, we agree. Humans aren't going extinct. Awesome. That also means genetic entropy is not happening, because the way Sanford, and I should note, Sanford coined the term and defined it in the book, Genetic Entropy and the Mystery of the Genome. So it is his term, it's his concept. If it no longer predicts the ultimate extinction of humans from genetic degeneration, then it bears no resemblance to the original idea that Sanford put forward. That's the whole point of it. It's, as I said in part one, it's an argument by contradiction, right? It's saying, if this is true, then evolutionary theory is false. And then he spends the whole book arguing that it is true. If he's now removing the extinction side of that, it takes away the therefore evolution is false part of that argument. So I'd be very surprised if he removed that part, but I may be wrong. Let's find out.
No, page 45. It's page 45 in the updated version. If Erika, it says here, if not rapidly corrected, this situation leads to the eventual death of the species. In the final stages, genomic deterioration leads to declining fertility, which curtails further selection.
Okay, the death of the species, that's extinction. So he has not changed that argument. He removed the word extinction, it sounded like, but if he's talking about the death of the species, the word for that is extinction, right?
Extinction. Yeah,
Okay.
I'm not saying that, but to say that, I just have to clarify something. Genetic deterioration can happen before extinction. That is how you know that we're headed toward it. That is how we'll know.
I agree, but then that goes back to the paper I just showed you, showing that no, it actually doesn't inevitably decline. And that paper, which is a model, but the parameters
by Warren Ewens, right?
Well, the parameters
No, that was a paper by Ewens.
Yeah.
Who uses the definition of fitness that is flawed.
I think that's going to be our next topic. So let me just say this about this paper. I'll give you the last word and then we can go into the definition of fitness. But so that paper, what that paper shows is that that paper actually uses parameters very similar to what Sanford describes, in that everybody is getting slammed with deleterious mutations in that model population. It's not comparing optimal to whatever. It's everybody is suboptimal in that model, and yet they're still able to be sustainable because you have selection. And the great thing about that model is that it actually matches experimental results that we've seen. Experiments in the lab with not just mutation accumulation, but mutation saturation, where a population experiences every mutation. That's the Springman 2010 paper. In that experiment, you see results that almost exactly match what that model proposes. So that, I think, that's it for the idea that whatever fraction of functionality, if you have just mutations accumulating, that necessarily leads to extinction. You have theorists that certainly say that, and Sanford puts it in a creationist framework. But here we have a model and data that match the model, and the data are actually from before the model was published. So it's not even like they're matching their experiment to the model. They're working independently from each other, and they reach the same conclusion. So I'll give you the last word on this topic, and then if you want, we could hop over to fitness.
Well, thank you. I'll try to go in the 20-minute. Those are very compelling objections. I'll point out why I think the problem with evolutionary definition of fitness is deeply flawed. Beneficial mutations can arise from loss of function. This is known. We call that reductive evolution. Michael Behe in the peer-reviewed journal it was Quarterly Review of Biology says, and even Jerry Coyne thought it was a great article, that most beneficial mutations are loss of function. These adaptations are loss of function. So neutral mutations, so beneficial mutations in the evolutionary sense are considered loss of function, can be considered, most of them are loss of function. Neutral mutations can be loss of function, and deleterious mutations can be loss of function. And that's why I said the fitness definition being used by evolutionary biologists is nonsense. So you picked out examples like sickle cell anemia, where the sickle cell allele is a loss of function. It will compromise the individual. There is a book, The Survival of the Sickest, where it says why we need disease, because it is so apparent that we have all these diseases. We have to characterize, we have to equivocate, we have to obfuscate that these losses of function are beneficial, because the paucity of actual functional increases where we can get new organs and new capabilities, such as complex new capabilities, like say a creature that didn't have the capability to sense electric fields. How does it acquire it? It requires so many simultaneous changes. So the easiest change for evolution is to knock out something, because there are many ways to break something, very few ways to actually make something. So that means most ways that mutations work will be that you can break something easier than you can make it. So what evolutionary biologists have done is to take all this breaking and claim that this is a beneficial mutation. And look, this is how things can evolve, by reduction. And there are more and more papers that talk about reductive evolution. Reductive evolution is where we have beneficial mutations just trashing an organism. And that's why I object to that definition. And when you say correct definition of fitness, it is not correct. It's the evolutionary definition, which makes it bad.
Dang, you guys are on it time-wise. That was exactly like 19 minutes and 55 seconds or something along those lines. I'm thrilled by this. I've got to do nothing. My job is easy. I will come and hunt down chat, though, because we've got 313 people watching and only 80 likes. So I'll hunt you down and ask you nicely, like the video. Do it for James. We all love James.
Yeah.
And with that, I think we'll jump into the next topic. What do you guys think?
Which I'm guessing is it going to be fitness? I hope it's fitness.
I don't know.
It's going to be the topic that I choose. If that was my understanding,
go ahead.
Dan, if that's all right.
Please.
Sal's taking the lead on this one.
Let me see. I'm trying to. Is my screen shared?
Yes, we can see what you got.
All right. So I'm showing a picture of God-made RAM. I call it God-made random access memory. And I'm going to show a video here. Hope this works.
In a recent study, we show that when the 2-meter-long...
Oh, did I blow that? I'm sorry.
We got booted. It's okay, Sal.
I got booted. Let me try this again. We're having a little technological problem. I appreciate your forbearance here. Try this again. Okay, share.
I'm watching that like number climb up. Good job, chat. You're spared for now.
Right.
In a recent study, we show that when the 2-meter-long human genome folds inside the microscopic nucleus of a cell, it forms 10,000 loops. Each of these loops connects 2 anchor points which are far apart along the chromosome, but which loop back, coming close together as the genome folds. In our new study, we find that loops form by a process called extrusion. Let's take a look at how this operates on a segment of DNA. First, an extrusion complex containing 2 subunits attaches to DNA, forming a small loop in the process. Next, the 2 subunits slide along DNA in opposite directions, making the loop grow bigger and bigger. The extrusion complex looks for a specific motif, a DNA word, which causes a protein called CTCF to bind to the DNA. If a subunit encounters a CTCF motif that is pointing at it, the subunit tends to stop. If the CTCF motif is pointing the other direction, the motif won't be recognized. To the subunit, the motif looks as though it is backwards. This behavior leads to a result we call the convergent rule. The pair of CTCF motifs at the ends of a loop must be pointing towards one another on the DNA. Our simulations also showed that when loops form by extrusion, they lead naturally to the formation of what we call loop domains. This means that if two pieces of the genome are inside the same loop, they tend to bump into each other more often. In this physical simulation, you can see how the extrusion of two loops leads to the formation of two spatially segregated domains. Like the convergent rule, the association between loops and domains was observed in our earlier study, but at the time we did not understand why this was the case. Both findings, and many others that you can read about in our paper, can be explained by the extrusion mechanism. In a recent study...
Okay, thank you for letting me show that video. We had some technical problems. So we're talking about these extrusion loops. This is how a lot of genome regulation is modeled now. And this is the subject of the 4D nucleome project. So I'm going to just model, this is my drawing of that extrusion loop. And this is going to relate to aspects of junk DNA. So that loop there has to have these CTCF binding motifs, and that's the consensus motif right there. Now, where do these binding motifs, where are they often found? By the way, I do some work on the topoisomerase II enzyme. They're found in signs and ERVs. There's literature on that. And so the gene might be only a small segment. The coding part might only be a small segment in this loop. But there's a lot of stuff where we have all these molecular machines that have to aggregate to be able to do regulation, because that's how you have differentiation of cell types. So the formation of these loops and where they are in the nucleus affects cellular differentiation. So there's something here with the Sternberg-Collins paradox, where it talks about how these transposons have to be positioned just right. I don't have time to cover it, but this is a depiction of the 4D nucleome. Here is the nucleus of the cell, and here is the outer part of the nucleus. And we have these things like we just talked about, these loops. That's what we have, one part. So the 4D nucleome says that a lot of gene regulation and a lot of the way things get transcribed, et cetera, has to be, it's dependent on the physical location and structure. Now there is gene regulation, that co-regulation that happens in between chromosomes. So we might have this, see like these intrachromosomal reactions. And I showed one with the hot air link RNA. That's an example of intrachromosomal reactions. So what is this telling you? If you're just cutting randomly these pieces of these loops here, or if you're putting the ERVs or transposons or whatever in the wrong place and they happen to have a CTCF binding motif, you're going to be changing the compartmentalization of all this. It can't. That's why when we have transposition mutations in people, there's a lot of bad stuff that happens. So it raises the question: how did this 4D nucleome emerge to be able to regulate in such an elegant and powerful way? It's not easy. We can't build machines that sophisticated. So here's an example with the fire link RNA. Link RNAs were considered junk at one time, but this link RNA is able to help bring together chromosome 2. I'll show close up: chromosome 2, chromosome 15, chromosome 17, and chromosome X. It's going to bring, so this was considered junk DNA, no longer. It brings these chromosomes into proximity and they're able to co-regulate each other. Here's another example of DNA that was thought to be junk. This is, that pink dot there is the X-cis link RNA. It does dosage compensation on the X chromosome. So that little pink thing is the X chromosome with the X-cis link RNA changing the chromatin state. So I showed these chromatin computers earlier. So great tribute to the people who are able to make these visualizations. There it is. The X-cis link RNA exploits three-dimensional genome architecture to spread across the X chromosome. If we think that most of the nucleus there and the DNA is junk, that does not agree with the idea that we have this computational 4D nucleome regulation that can happen in 3 dimensions and regulate. By the way, these what they were showing is there, and it's really hard to depict this. Well, let me move to the next slide. These associations change depending on cell type. So we have at least 300 canonical cell types, depending on how we count them. But this has to change so that you have when you have a nerve cell, you have versus a bone cell, these interactions change, and they depend on this, what was called junk DNA. We're finding this out more. What happens, by the way, if we disrupt some of this at the nuclear, just a little bit at some of the DNA, the areas here? Well, we have a condition called progeria, premature aging. The life expectancy is around age 13. And this is from a biology textbook. Defects in a particular nuclear lamin are associated with certain types of progeria, rare disorders that cause affected individuals to age prematurely. Children with progeria have wrinkled skin, lose their teeth and hair, and often develop severe cardiovascular disease by the time they reach their teens. So what am I trying to prove here? Structure, structure, structure. If this structure is just junk, it's not going to work. And even just a little bit of perturbation of that causes some bad things to happen. We're finding that out. That's why we're studying it. That's why medical researchers think a lot of this is functional. It's their job to find out what causes disease. And they're being told by evolutionary biologists that this is junk. And the data does not agree with that. So let me go to some specific examples here. Some things that were considered junk, like line transposons, which are about 17% of the genome, give or take. We have the line one. This is from a bioarchive, and I'll show even more. This is what they had to say. By the way, I'm sorry, I had a little bit of a mishap there. This is an example of a line one, and it says dynamic expression. This is like 2020. So some of the stuff you may be hearing about this being junk, it may be an obsolete argument by now. So from this paper, dynamic expression and essential roles of line one in mammalian development. Line one is a transposon considered junk, used to be. Line one is highly expressed during several stages of mouse and human development, notably during the global reprogramming of DNA methylation and histone modifications that take place during pre-implantation and germline development. The study of one such context, pre-implantation development, has revealed essential functions for line one RNA in regulating both rRNA and two-cell specific gene expression by promoting recruitment of key factors to chromatin. Line one expression also impacts global chromatin organization via direct or indirect mechanisms that remain to be elucidated. In separate studies, line one expression and mobility has been linked to neurogenesis. So that's one example. Now I'm going
9.5 minutes there, Sal, just to let you know on the time.
I'm sorry?
I said you're at about 9.5 minutes there, Sal, just to let you know on the time. I paused it for talking. So
okay,
sorry.
I'm gonna, Dan, I'd like to continue if that's all right with you, because I'm trying to answer your questions here about.
Totally up to you guys.
I mean, I'd like to get a word in because I have
go ahead, have
questions
a word.
about what the argument is here, because this is starting to
the
sound.
The argument is you posted that only 11% is functional, and I'm trying to contest that you may be wrong. You may be dead wrong.
What I said specifically is that we have strong evidence of functionality for 11%. We also have somewhere in the neighborhood of 10% that we're not sure about. And I think the best conclusion is that the actual functional fraction is in the neighborhood of 15%. It might be as high as 20, but I'm not as convinced of that. But I think 11% is what we can be really confident about right now. And I think it's reasonable to say that maybe up to 15.
So my argument is I'm just trying to show that all this stuff that has been saying that ERVs and transposons are junk, that's an open question, and I'm trying to show the reasons why. And I think it's important people see this, because I don't want evolutionary biologists to keep getting away with saying that ALUs and transposons, other transposons, and ERVs are junk. I'm trying to show the functionality.
I'll defer to Dan here because you guys agreed on the time for 10 minutes. So if he's cool with you going over 10, that's totally up to you.
Well, I'd like to get in if that's okay, because I I agree with some of what you're saying, and then I think you take it several bridges too far. Well, here, can we stop the share?
And if Sal wants to finish with his 30 seconds and see what else he wants to get,
go ahead. Yeah,
by all means.
Okay.
And then we'll quit the share.
All right, 30 seconds. Thank you.
Yeah, no problem.
So Ayala was saying that eludes were junk, and that has been falsified on many levels. He can't categorically, unequivocally say that. It's an open question, but there is a lot of evidence. Here it is. Just all these potential things of function, not just potential, but confirmed where there's association. So I don't, so this whole thing that evolutionary biologists came up with, that's saying the genome's junk, that's an evolutionary biology thing. That's not based on experiment. This is based on experiment, what I'm showing here. So Dan, go ahead. Thank you for giving me a little bit extra, Dan.
Oh, no, all up to you guys. I'm just trying to.
I will stop sharing my screen.
thank you. So, okay, so the argument here is that and I want to just reiterate just to make sure I get it. The argument is that a lot of the genome is functional. Therefore, it is very tightly constrained. So if we have mutations, because so much of it has some important function, as it mutates, stuff breaks and we degenerate. Is that a fair characterization?
It has the chance. Yes, that's a fair characterization. But also the other thing I'm trying to point out, this is an example where evolutionary biologists got it wrong. They're not dealing with the facts. They're dealing with theory. This is an example. This one molecular biologist said this whole thing about junk DNA was the greatest mistake in the history of molecular biology. So I'm highlighting this. I'm trying to set things right because there's still a lot of this where you say you think 11% is functional. No one knows the figure. No one knows the figure, but I'm trying to give reasons why it may be different. But yes, there are a lot of people that do think.
Oh, did we lose Sal?
Sal, we might have lost you. Sal's making a funny face, and I don't think he's joking around.
I think he's mocking you, Dan. But let's see. We will wait for him to come back.
Sal, come back. He's probably still talking.
It's like in the movie Suburban Commando when you could get frozen by the freeze gun.
It's just like that, James. It's just like that.
You ever seen that movie with Hulk Hogan? Okay.
I would never miss a Hulk Hogan movie. Surely you know this about me by now.
It was pretty epic. Are you serious?
Sorry.
Okay.
I wish now I feel like I feel bad.
Last time we do this, Erika. Well, we lost
Sal's
Sal.
gone, but he'll be back.
He's gonna hop
Sal.
He'll hop
I'll wait for Sal.
I'm glad it's not my Wi-Fi. Usually I'm the one that's having to apologize for roboting out.
Don't worry. Wait, I think you just got Sal back.
I reconnected on another line.
Okay, sweet. All right.
So I'm back. So where was I? About a minute ago.
Biologists were wrong because more of the genome is not junk than they originally
Evolutionary
said.
Biologists were wrong. It wasn't the view of some molecular biologists. And I was saying, notwithstanding the fact that you cited Ewens and you're saying Grauer is wrong, there is another very respected population geneticist, Joe Felsenstein, that is drawing the line in the sand and saying it has to be junk. You can see that in his textbook, graduate level textbook, version 2017, Evolutionary Theoretical Genetics, and just do a search in that book. It's freely available online, and just search ENCODE. You'll see his opinion. So notwithstanding that you cited some papers that will say that fitness will increase under all dysfunction, that's not the universal view of evolutionary biologists on the whole. That may actually be, I can't say it's, whether what you cited is the minority view or not, but they're, Felsenstein and Michael Lynch are pretty respected people. So.
I hear your argument. First thing is that saying these smart guys agree with me is not an argument. That's not how being right or wrong works. That's an argument from authority saying these well-respected geneticists say this. Well, that's fine, but do you have data to back it up? What I'm saying is we have new information. We have new papers. I've shown you so far tonight 2 papers from 2020 that back up my position. So I think that's the first thing that needs to be said. The second thing is let's look at the overall genome, because when these original estimates about junk DNA, which is not a term evolutionary biologists, really any biologists use anymore, when this term was invented, we didn't know what was in the human genome. It was actually, as you showed, it was before we sequenced the human genome. We didn't actually know what the contents were. Now we know most of what the human genome is. Like I said, there's maybe 10-ish% that's not well characterized, but the rest of it we know really well. And you showed that slide earlier that I actually showed last time that kind of broke down the broad categories. Now, I wanted to say, well, hang on a second. You mentioned a few specific things, like you mentioned ALUs. Okay, ALUs make up about 10% of the genome. Is anybody proposing that every ALU repeat is functional?
No.
Not to my knowledge. I know there are a few that have been identified as having some physiological role, but nobody's saying anywhere near that full 10% is going to be functional. As far as I can tell, it's the ones that are functional are very much the exceptions. Is correct? Is that an appropriate impression based on what your research into the topic?
We don't know, but 95% of adenosine to inosine editing is done in mammalian organisms is in the ALUs. If we shut down the ADAR enzyme, the creature dies or it has just brain seizures. So we don't know. But the thing that I get pretty upset,
we don't know.
Evolutionary biologists like Ono will say it's junk, even before we even know what's in the
and that was back in what year,
example,
though?
where evolutionary biologists speak first, don't look at the facts. They look at the facts second and only reluctantly begin to back down when the facts are just undeniable.
What year was that?
That's what I was highlighting.
That determination made. What year was that? That was what, in the 1980s? You showed it before. I forget the year. What was the year?
1972.
1972, before we had ever sequenced any genome at all, even that tiny little phage, 5X174, right? So now it's 2020,
I thoroughly
and we
agree.
know what's at Sal.
They come out and talk first, make their theories appear like facts, and then the facts come and run them over. That's what happened. That's why this is an embarrassment. You know, it would have been better to just say, we don't know. We're looking, than making these declarations of how the genome works just based on evolutionary theory and not on empirical evidence.
Okay, Sal, that is how science works. We take our shot.
That is not how science works.
Evolutionary
theory is not science.
And if it turns out that
that's
it's
a good
wrong,
example,
then we correct it. That is how science works. And so let's look at what we know in 2020 instead of 1972 about the human genome. And you showed the slide here. I'll pull it up right here. I'll pull up a little bit right here. So the first thing
I'd like you to have, please, Erika, it's okay. I'd like to give Dan the last word. He's been very courteous. So I interrupted him a few times. So
no, you guys are still good on time. You guys have about 5 minutes left.
Yeah. So this will
the last word, Dan. I'd like to defer to you. You've been very courteous and civil. So
thank you. This will not take the full 5 minutes. This will be pretty quick, I think.
Absolutely.
So what I want to show you here is, first let's look at this. So this is a breakdown of the human genome right now. And I showed this last time. I'm not going to go through everything, but basically we know what it is, right? We know transposable elements, mostly retrotransposons. We know endogenous retroviruses, the remnants of viral infections. There are about 1% pseudogenes. They're just broken. Within all this stuff, there is stuff that is functional. So for example, within introns, there are enhancers. And that's actually the reason for the lactase persistence. It's an enhancer in an adjacent gene to the lactase gene that mutates to become more efficient. It's within an intron of that adjacent gene. So like the part that's introns that we're saying is non-functional, we're saying it's stuff that we know is not something like that. Now, if you dig into these numbers more, you can get more specific. So for example, we have about 10% of the genome that's ALUs. Nobody is saying that all 10%, that full fraction, is functional. Maybe it's 1/10 of it, maybe it's half of it. Sal's right when we say we don't really know. What we do know is that it's not the full 10%. Nobody is making that claim. And you can do that bit by bit through things that creationists claim are functional and say, well, here's an example that's functional. Therefore, this whole class, or implying that we should consider this whole class functional. And that's wrong. So another example of this is with endogenous retroviruses, which are about 8% of the genome. It is absolutely true that some of the transcription start sites for endogenous retroviruses are now really important for human gene expression. We've actually, where those viruses landed in the genome, we've dealt with using their components as part of our own regulatory network. But that's very much the exception. And you could find papers that show that. And Sal probably has a couple of them. But for every ERV that has a functional promoter, there's dozens, hundreds that are so degenerated that they don't have that. Plus, even if you were to look at the ERV, the promoter is a teensy little fraction of it. If it's intact enough to have an intact promoter, you have to look at the rest of that endogenous retrovirus. It's got 3 main genes, GAG, POL, and EMV. Those don't do anything for humans. They're actually, if they are expressed, and there have been experiments on this in mammals, if you mess with an ERV to allow the expression of those proteins, they actually cause a disease. So they're very much not functional. To the extent that there would be part of that 8% functional, it's a minuscule part because promoters are tiny. We're not talking 1%. We're talking maybe we're not talking 1/8 of that, like 1%. We'd be talking like 1% of that 1%, right? It's a teensy tiny fraction. So you can go through we have all these functions that we're finding in the genome. 100%, I'm not going to disagree. But along the way of finding all these functions, we're finding a whole lot of stuff that we now know what it does. And the answer is either nothing or it hurts you. So that stuff cannot be considered functional. And there's just one more little thing I want to show you here. It's this figure, which is from Kellis 2014, that shows, this is probably hard to read for everybody, but what this shows, and this is kind of a follow-on to ENCODE, is this shows genome biochemical activity broken down in a bunch of different ways. So like, for example, the red circle is the fraction that is conservation, and that's between 8 and 15% of the genome is evolutionarily conserved. That's a strong argument for functionality, I think. This little purple circle, that's protein encoding. That's like 1.5-ish%. This green oval generates a phenotype. So all that stuff Sal's talking about like, well, this thing is associated with this disease. Yeah, those things exist, but it's not a big fraction of the genome. There's just no way to put together all we know about the genome and all the different functions and constraints in the genome and get more than like 20 or maybe on the outside 25% functional. It's very possible that in 10 years, I will be completely wrong about that. But based on what we know now in 2020, not 1972, not 2005 with the original ENCODE paper or whatever, 2004, whatever it was, but right now in 2020, we know stuff, and it's not 80% functional, it's not 50% functional, it's maybe 20% functional. And that's, I think, where I think that wraps up for that segment.
Dan, disappointing. That was 20 minutes rather, and 2 seconds. So you were 2 minutes over.
Okay, Sal, that one was you, right? So we're back to me?
Yep, it's your segment.
Is that right?
Yep.
Oh,
go ahead.
Okay. All right. So and just point of order for James, I'm assuming this will be the last one, and then we'll probably go to questions. Is that right? Just based on the timing?
I think that would work best. Yes.
Can everybody hear me?
Yes.
Sorry, I cut out a little bit there, but it sounded like it's probably another 20 minutes, and then because. I mean, I can't speak for Sal, but I'd be happy to do another 3 rounds, but we probably don't want to keep people here that long.
A little longer than I expected, but if it was in the email, it's my fault for missing it.
Sorry. Okay.
It's my fault.
I'm fine. Dan, maybe I'd like
let's do one more. Let's do a real quick one. Let's do let's say 10 minutes, because I don't have like presentation. Let's just do like 10 minutes and then we'll call it good. Okay. So final topic that I really want to get into here is fitness, because this is like the elephant in the room. We have to figure out what the right definition of fitness is. And the answer to that is the evolutionary definition of fitness is the right definition of fitness. And I'm going to talk about why. So here we go. I'm going to share this, but I swear, this is very short. This is not like a 10-minute thing. Okay. So look, last time, Sal, you said you're worried. You said I think it was at the beginning of your rebuttal, you said, you're worried about the future of humanity. And a little while ago, we went back through Sanford and we agreed that according to Sanford, the ultimate outcome of genetic entropy is extinction. So you can ignore the if no, because we agreed. But if the answer is yes, and we agree the ultimate outcome is extinction, then we agree on the definition of fitness. XYZ, something, something, something happens, and then eventually reproductive output declines and extinction results. That's what we're talking about here in the context of genetic entropy. And in the context of a population being viable, in other words, a population not going extinct, if you experience a mutation, and I want everyone to listen to this because it's counterintuitive, but listen to this carefully. If you experience a mutation that increases your reproductive output by 1,000%, okay, you increase your reproductive output by 1,000%, but 90% infant mortality rate, that's still an enormous increase in the viability of that population on net, even if in terms of a health-based or a physiology-based definition of fitness, some non-evolutionary definition of fitness, that is a huge decline in fitness. But in terms of the question of extinction, that hypothetical example, which by the way is completely unrealistic, it doesn't exist, but that hypothetical example is an increase in fitness in that population with that new allele will be safer from extinction than it was before that mutation occurred. The XYZ in this formulation does not matter because everything, whether it's intelligent or
One sec.
whatever thing you
hold
come
on.
up with, structural
Dan,
precision,
we
nucleus.
weren't quite able to hear you. I think for maybe the
Robotic.
last like 5 seconds.
Oh, so the last 5 seconds, sorry about that. The last 5 seconds were that the XYZ in that formulation doesn't matter. Whatever it is, whether it's structural constraints in the nucleus or health of the organism as an adult, all of that is going to be downstream of the genome, downstream of the genome sequence, and upstream of reproductive output. And again, I want to make this very clear. This is all about reproductive output. So you can talk about mutations or, like Sanford describes, information loss, the 4D nucleosome. It's all about, on one end, you've got changes to the DNA. And on the other end, you've got changes to reproductive output. And the question, when it comes to will a population go extinct, is will that reproductive output stay the same, go up, or go down? That's the relevant question. And this actually is what we've been talking about, even though we're using different terms for it, because Sal, you have this thing you call the bonkers equation, which is for everybody, we didn't use the words tonight, but it's the table that Sal showed earlier, where it's like if you have X number of mutations or X% of harmful mutations, you need Y number of offspring to be viable generation over generation. If the problem that equation describes is that each person must have so many offspring for the population to persist, then we agree that the relevant question is reproductive output. The problem isn't inherently that population is less smart or less fast or whatever. The problem is that they're not having enough kids, right? And that is using, whether you want to use it like you want to say the words or not, that's using the evolutionary definition of fitness. Now, the implications in terms of the actual discussion we're having here is that if you're insisting on a new definition, creationists are acknowledging that they can't win in the terms of evolutionary biology using the correct evolutionary definition. And I'll give you a specific example of this, and this is where I'll leave this. And Sally, you can have the last word here. I'll leave this with a piece by Paul Price and Robert Carter on creation.com. That's CMI, is the Creation Ministries International, called Fitness and Reductive Evolution. And that's Rob Carter, that's Dr. Rob Carter, that's Dr. Sanford's co-author on that H1N1 paper that they claim to show genetic entropy and don't for about half dozen reasons. But in this piece, arguing basically the same case that Sal is making here, they say in the conclusion, only when we insist that the terms of the debate be fair and accurate will we have any chance to clearly communicate the truth of creation and the bankruptcy of Darwinism to the world at large. And that right there is saying the quiet part loud. It's saying we can't win in the context of evolutionary theory. So we need to redefine the terms of the debate to our liking, and then we can compete. But if you use those words, it doesn't work. So that's why we need to have the redefinition. And I'm good. So Sal, if you want to have the last word on that, it's all yours.
Oh, thank
Got
you.
like
Thank you.
5 minutes if you want to use the rest of the time or banter or whatever you guys feel comfortable doing. Just go ahead whenever you're
Okay,
ready.
5 minutes. The notion of fitness, in the common sense notion, a submarine is fit to operate under sea. A space shuttle is fit to operate in space. An airplane is fit to operate in the air. A car is fit to operate on the ground. These are common sense notion of what it means to be fit. And the question of how the complexity of life and it's able to do these wonderful things like an eye is able to see, it's fit to see. These are common sense notions. These are not creationist notions. These are our notions from our understanding of how machines and engineering works. These are common sense notions of medical. These are medical notions. So it's really bad when we have a situation, and Alan Orr alluded to this, where we have a blind crustacean, like say Gomeris minus, and he made a very good critique against Daniel Dennett. It was a beautiful quote where he said, selection's able to destroy design as much as it is able to build it, though I would disagree that it can build it very well. It can certainly destroy it. So natural selection can destroy designs. And he said, natural selection does not trade in the currency of design. So it's not, I think, very honest. I don't, I hate to use the word dishonest because I think this is just delusional. Dishonest means someone knows it's wrong and then they're going to say the wrong thing anyway. So I don't want to, just make something, be very pejorative here, but a lot of the definition of fitness ends up being just obfuscation. So when we're talking like in some of these experiments where we saw all these beneficial mutations coming in, it could be just damage to the promoter region where the gene shut off. There's so many ways to break a promoter. There's so many ways to break a gene, hundreds of ways. You can throw these stop codons and have a protein truncation variant in thousands of locations of that of a big gene. And if it causes the creature to reproduce more, like say in the case of Gomeris minus, where it's in a cave and it's blind, it's counted as beneficial. I think that's, that's fine as far as evolutionary definitions, but they can't use, and what I'm just doing is I'm calling out, you can't use the word beneficial in those cases where we've had loss of function and pretend this is proof of evolutionary theory that it can build complexity, build integrated systems, because, what is the mechanisms that make common ancestry possible? So it used to be believed that it was the product of natural selection and mutation, but there are a lot of people that accept universal common ancestry that now have their doubts whether natural selection plus kind of random mutation is adequate. And these are some open questions. And I'm just calling out the problem is if we're going to call reductive evolution an increase in fitness where Gomeris minus loses eyes or winged creatures loses the ability to fly, then we can, it's very easy to say, hey, look at all these beneficial mutations that are coming up. But then it really doesn't answer the question of how complex organs and systems come up. So this is why I'm criticizing the idea of fitness. It's not just the creationists who pointed this out. Richard Lewontin gave up on the idea. And then there are I have papers here by Brady and then by Grodwall and Andreas Wagner. So this is a serious problem. If we're going to try to model, if people are going to claim the evolution of complexity through natural selection, they better define it in terms of complexity in the engineering sense. The other thing is another emerging discipline, and I know I have about 1 minute, another emerging discipline in biology is biophysics. The biophysicists don't define kind of this idea of functionality in terms of reproductive success. They define it in terms of machines operating at the limits of physics. When a machine operates at the limits of physics, it is maximally fit. In some other discussions, I showed the maximal fit in operation, the optimality of an ion channel. It has to have sub-angstrom precision. That just makes sense. Nuts and bolts have to fit together correctly. That's a nice metaphor for the engineering notion of fit. The evolutionary biology definition is just not appropriate to the questions at hand. So thank you very much.
These guys are pros, you guys. That was also just perfectly on time. Look at, I barely had to do anything. All right. Good job, everyone. I had a ball listening to all that, and I'm sure everyone out there in the internet at large did as well. But the fun's not over yet, because we're gonna do some super chats, and I'm gonna try to.
Before we
yes.
get into, are we doing closings before or after questions?
Oh God, James, sometimes I roast you in my head for forgetting about these things, but then I forget them too. Yes, you guys did have closings planned, and I think we typically do them before we do the super chat. So if you guys want to do closings, Sal, since you started us off, would you prefer finishing first, or would you like to go second?
I was expecting to do closings at the end, if that'd be okay with Dan.
Yeah, if that's okay with
It's
you.
okay with me. Yeah, sounds good.
Yeah,
So we'll do the super. We'll bring in some money first, okay? So
okay. All right. Yeah, we gotta line James's pockets here. He's the big business fat cat on this one, right, James? Making all the big bucks.
That's funny. I have to make an announcement about the channel. I am super excited and thankful as when I went in to check on the super chats, we just broke 25,000 subscribers. So I want to say thanks so much, folks. I am so thrilled. The debaters are the people that I have to pass on the congrats and thanks to, and just say we are excited about the future. And so thanks to guys and gals like Dr. Dan, Sal, Erika, and others. So really excited about that. And I will kick it back to Erika, but I just saw that and I got excited. So
James, people come here for your superb moderation. I'm the consolation prize. Everyone was hoping you were moderating.
that's funny.
So I can start us off though with these super chats. So first of all, from Logical, Plausible, Probable, live after show, looking for link, and I believe that's on Standing for Truth's channel, if I remember seeing it in the side chat correctly. So just pop into your YouTube search bar and put Standing for Truth, and you'll find their after show. They're hosting one over there. From Ryuzinski for $5. Oh, sorry, Logical, Plausible, Probable.
So 1 second. Forgive me for interrupting, but just wanted to mention that these are actually super chats. I sent Erika a document, and I think I didn't tell you this. Sorry about that, Erika. These are super chats from the last debate that Dr. Dan and Sal had. So we ran out of time.
Catching up.
And what happened was when we did run out of time, I told people, I said, I'm going to save your super chats if you sent a super chat, because we do want to read those. We want to work hard to get those in. And so these are actually from there's only about maybe a handful of them that we didn't get to ask from the last one. So if that's okay with you guys, we want to, and if you're kind of like, boy, I haven't read that for a while, that's totally okay to say that. But we thought better late than never.
Man, James, you made me look like a dingus, or maybe I just didn't read your email all the way. I saw super chat and then I just clicked and I got overexcited.
But though he is actually having an after show tonight anyway.
Yeah, that's what I was gonna say. I was like, okay, well, but saved, I guess. But from last time, Ryuzinski for $5 to both debaters, do you think you or your opponent is more qualified to argue this topic? Thank you to both of y'all for appearing. Sorry, James. I don't want to know what that apology was for, James.
I don't have an opinion, but I think Dan's very excellent professor. He's very knowledgeable, and he's definitely everyone has said he's been a worthy opponent of me. So, or I'd like to think I'm a worthy opponent of him. So that's all I have to say on that.
Yeah, my 2 cents is that it doesn't matter because qualifications are overrated. It's about the arguments you're making. It doesn't matter who has what letters after their name.
Cool. Yeah, that's, we screen interlocutors. James and I screen the interlocutors to find the diamonds in the rough like you guys. So from Dave Gar, last time for $5 for both, please discuss apoptosis, which for those of you out in the audience is controlled cell death, in the context of genetic entropy. Micro level stuff here, not at population level.
I'm happy to take it, or you can take it. Either way, I mean, we'll just.
I don't think I'm qualified to comment. I mean, it's an excellent question, and I'm sorry I can't give you your money's worth for that one. I also have to turn down the air conditioner, so Dan, you got it.
All right, I'll give you a quick one here, and I apologize because I'm going to take a micro level thing and turn it into a population thing. I'm going to set myself a timer here, see if I can do this in 1 minute. So the way I want everyone to think of, so apoptosis, programmed cell death, in the context of a multicellular organism, you have cells that go through a process of death in such a way that doesn't hurt the surrounding cells. And this is the thing they decide to do. It's not personifying them, but you know what I mean. And this is hard to explain evolutionarily, but I want you to think of the human body not as one entity, but as a population of trillions of cells that are all genetically identical. So if you have an allele that makes it more likely to have offspring, then that allele will, in theory, be passed on as long as it's present in the appropriate cells in your body, and they're all identical. So an allele for apoptosis seems counterintuitive because that cell dies. But if it helps the whole population do better, which is to say reproduce, then that allele is going to propagate and apoptosis will be selected for. It's counterintuitive, but it works really well.
All right, sweet. Okay, next up, Sal, this one's for you from Sigifredo Sarabia. For $10 US at Sal, does DNA carry a template or information to create, or does DNA pick up the information along the way? When and how did the information get there, if not by evolutionary processes? Spoken maybe, question mark, or question mark? I'm not sure. I mean, I guess these things spoken into being, perhaps, and then...
I may have to ask you to read part of the question again, but I'll answer the last part of the question. How do I think information came to be? I think a miracle did it. There are a couple models of creationism where a miracle, we have like progressive creationism and then young life creationism, young earth creationism. So he asked what I believe. I believe that about 6,500 years ago, there was a miracle that basically created all life forms. So how does DNA acquire? It acquired most of the information at that first event. Can we prove it scientifically? I think that is outside of science. The reason I believe that the information came, and I don't like to use the word information, but the idea, the information, the configuration of DNA, that it came about by a miracle is the problem of abiogenesis, which is separate from evolutionary theory, is very, very substantial. I'm more convinced of a miracle involved in the first life. The reason that I think life popped up fully formed is the evolutionary transitions seem to be requiring miraculous changes, particularly the change from prokaryotes to eukaryotes. That's just enormous. And I know I mentioned Changed Hand earlier. When she started teaching cellular biology, she'd been an atheist, she became a Christian. When she had to teach cellular biology and she was highlighting, teaching the differences between prokaryotes and eukaryotes, that made her a creationist. It was just too hard to believe this could happen. So I don't know if she became a progressive creationist first, but she's a young earth creationist like me now. So what was the rest of the question? What was the first part of the question?
Yeah, sure. I actually think you kind of answered it. It says, does DNA carry a template or information to create, or does DNA pick up information along the way?
Yeah, it doesn't pick up that much information along the way. I do think there is some adaptability in the DNA. There is some intelligence to the way it's constructed so that it can adapt to an environment, and we have developmental plasticity that way, but not very much. So thank you for the question. That was a good one.
Gotcha. All right. And from Stupid Whore Energy, as she likes to be called, for $10 US, if you find transposons in the same place in the genome, it points to common descent because of all the places that they could have jumped to. Some of them get co-opted to perform some function. So what? So I believe it's not directed to anyone in particular. So I'm just going to let you guys both throw your 2 cents in if you would like.
I think that would be for Sal, because my answer to that is yes.
Okay. Yeah, I don't know if you wanted to comment or what. If it's,
I'll just briefly add that there's additional evidence for it. And it's that we can look at lineage-specific mutations that occur once they're integrated. So like, for example, you can look at insertions that are in humans and chimps that have mutations, where humans, chimps, and gorillas share the same insertion, but within that transposon, humans and chimps share mutations that gorillas don't have. So there's actually another layer of evidence for common ancestry in that regard.
I think one way to resolve this question, and this is one reason I'm interested in young earth creations. By the way, I used to believe in evolution, also in old earth, the old universe. One way that we can the question could be settled is we're able to establish that the fossil record is young. I think that'll settle it. So trying to just go after, in the absence of evidence, we're just arguing in the vacuum.
Pardon my interjection, but we probably have to move quick as we are going on. We're at 2 hours and about 2 hours flat. So to get through as many questions as possible, we might have to go through as fast as possible.
Lightning round, 30
both
seconds.
In terms of us reading them, and then also, I hate to ask, but just because we've gone for a good amount of time, maybe short and pithy responses, those usual strong punches and jabs that you throw, both Dr. Dan and Sal.
Yeah, they're the
Lightning
experts.
round, let's do it.
All right, James, I think you have the rest of the super chats. I don't have them in the email.
Gotcha. Okay, well, I'm going to read through. I'm going to send Erika that list as that caught me off guard. And I will also say one thing I just noticed. And Subatomic and Nano, thanks for subscribing. Saw you pop on the screen. Welcome to the community. I think there might have been some others I missed, but it's been flying fast. And so basically the basic idea, no matter what walk of life you're from, creationist, evolutionist, you name it, we hope you feel welcome here. And a quick reminder, all of our guests are linked in the description. So if you want to hear more from them, you can by clicking on those links. Now, I'm going to start reading. And as I ask this first question and we hear it back, I basically will send that list of questions over to Erika, and then we will start taking turns reading back and forth. But Stupid Whore Energy strikes again. She says Sal should check a paper by Lang et al., Science 2000, which provides structural evidence that the 8-barrel proteins, a widespread and complex fold aroused by a process of gene duplication and fusion.
I'm not familiar, but thank you, Stupid Whore Energy, for the reference. I'll look into it. Thank you.
Gotcha. Well, I will ask the next one then, just because that didn't give me enough time to send the thing to Erika. But
You said pithy, James.
Stupid Whore Energy, thanks for your next
Thank you.
objection. Says, what do you think of the fact that the nucleosome, touche, that's a good point. What do you think of the fact that the nucleosome was lost in the Dinoflagellata
Dinoflagellates.
phylum, and that the origin of the 4D genome can be traced back to the prokaryotes, according to the work by Creamer et al.?
That's my thoughts is that's based on circular reasoning, that evolutionary theory is correct to begin with. So that may be all false. Thanks, Stupid Whore Energy.
Well, she strikes yet again. She says, this time, does Sal have evidence that ALU elements shared by humans and chimps are part of the tiny minority that are within 3 UTR of protein coding genes?
No.
Gotcha. Next.
Pithy.
I have to give you credit, Sally. Nobody is more short and pithy in their responses.
Well, thanks, Stupid Whore Energy, for your support of this channel. I'm sorry I can't give you your money's worth tonight.
I am impressed by how pithy you are. Oh, wait. Okay. So I'm going to send these to Erika. Then do want to say, very excited, as we had a congrats come in. And so this one is from, who is that from? Let me just quick find this. Give me 1 second, folks. Sorry, I'm working on an old computer here.
It's okay, James. You couldn't do anything to get on our bad side.
Bless your heart. Okay, next. Stupid Whore Energy strikes again. Says, Eric and Kent, congratulations. Really special. Very exciting for you, Erika. Next up, I am going to type in Erika's name. And Erika, I have just sent that list of questions that we can alternate. Starting from the bottom, we'll go from there.
Gotcha.
Converse contender. Huh?
I said I'm refreshing with bated breath over here.
You got it. I also sent you the congratulations picture. So, and Converse contender, thanks for your super chat. Says, gosh, I heard James was born on Mount Olympus. Not confirmed, though. Thank you very much, Converse. He's going to be moderating tomorrow. And I got to tell you, folks, tomorrow we are going to make a big announcement at the start of that debate. So do tune in for that. And Speed of Sound of Gravity, thanks for your quote. Said, James, I was on my way to bail my mom out of jail with this money, but you were just too cool. Appreciate that, Speed of Sound. Next, we'll kick it over to, if you have the list, Erika, Godless Recovery is next, just to, as we're working our way up.
Yeah, I'm with you.
Bet.
I think this one goes to everyone, or maybe Sal. It says, listening to Creationists, facepalm emoji ×3. So they're coming at you, Sal.
No, thank you for the super chat.
It was for $2. I didn't mention that.
That's
For how much?
$2.
Okay. That's a $2 answer.
Sal approves. He's like, oh, okay, you know, all right. But
If I can humiliate myself for James, I'll do it. So there you go.
That's kind of you, Sal.
I might get from chat. Most people in chat would.
Appreciate that. Bless your hearts. And Nathan Foster, thanks for your super chat. Said, it's called evolution theory for a reason. Why is Dr. Dan here arguing a theory which, if it had evidence for it, it would not be called a theory?
Thank you for the question. So this is a common misconception about science as a whole. So in science, a theory is an idea that has the highest level of empirical support. It's an idea that is robustly supported by multiple lines of evidence, and it makes accurate predictions. It explains observations. So a theory in science is different from the word theory in everyday language, where it's just a shot in the dark. A theory in science is as close as you can get to certain. Some other theories are things like germ theory, theory of relativity. Those are all theories in the context of science.
You bet. Thank you.
Very nice, Dan. Okay, from Dwayne Burke for £2. James and Dan are twins that were separated at birth. So he's hitting us with the juicy gossip.
We do, and on the screen, Tom Jump, Dr. Dan, and I are triplets. As I always say, Tom and I are twins as well. We do, if you look at us, hold on, let me get in position. Wait, there. Twins! Okay, next.
Now I have to pull this up and see what we're I don't.
Alec McKinsey, thanks for subscribing. We are pumped to have you, as long as you're not a beta. Okay, I just really want to say beta tonight. Next up, Erika, the floor is yours.
Well, I think, wait, okay. I have logical, plausible, probable next on my list. Is that next on your list or no?
Negative. I have Nathan,
Okay.
wait, I have Dwayne Burke, I think. P.S. We're totally kidding, Alec McKenzie. If you're a beta, you're welcome here. And
All betas are welcome here. All betas are.
But wait, so you have the list that I just sent, right?
Yeah, I've got David Burke followed by LPP and then Stupid Horror Energy twice.
Okay. So,
Foster again.
Oh gosh, no, I'm so sorry. I got us lost.
It's okay, James. You want to finish this off here?
Logical, plausible, probable. Thank you for your super chat. Said after show on Standing for Truth's channel tonight, open mic. So that should be exciting on your brother Standing for Truth's channel, Erika. Are you excited?
No, I'm always excited to hear from Standing. Standing and I are buds.
Excellent.
Okay, from Stupid Horror Energy, as she loves to be called, for $10. Sal should check a paper by
One
laying
second.
it out.
So
Thank you,
sorry.
Senator.
I already read through all of hers.
Oh, we're good then. Okay, well, you read through Nathan Foster's other one as well?
No, we didn't actually read that one.
Okay, then from Nathan Foster for £2. Dwayne Burke's beard helped sink the Titanic.
Very nice. Congrats, Dwayne. Next up, thanks for your super chat, and want to say, appreciate all of your support. Taliesin Oberlander, you've been with us a long time. Said, James, you were supposed to screen my interlocutor. Very nice reference to Darth Dawkins. I've tried to recruit Darth Dawkins to come back. I said, the people want you back, Darth. No response, but we'll try for you. And
He doesn't want to hear you giggling like a schoolgirl, though, James.
That's true. He got sick of it. And there's only a few more super chats, so I'm just going to quick read.
You read them, James. You read them, because I don't have them.
Corporal Annan, thanks for yours. Said, Dan, I love you. you. Okay, and jungle jargon. Dan's just as speechless as I am. Jungle jargon, thanks for your question. Said, ask Dan, how can you build an organ from mistakes?
So I will first, and I'm going to set a timer 1 minute. First, I object to the classification of random mutations as mistakes because they are just random events, but that doesn't hold a value judgment with it. So I wouldn't call them mistakes. They're unintentional, but I don't think that implies mistake. But the answer is that you don't build it all at once. You have little pieces that do little things, and we can see this developmentally, for example, in things like lungs, where it starts as just a little fold in the digestive system, and that's folded already for other reasons that we're not going to get into, and you end up with an air-filled sac, and fish kind of gulp down air, and that's called a swim bladder. And if they can pull air from that a little bit, that helps them get more oxygen into their tissues. So that's useful, just to give you an example. And then developmentally, it's really easy to tweak how these things form because genes that regulate developmental processes aren't saying like, make a lung. They're saying fold, pinch off, fold inwards, fold outwards, extend. They have very specific kind of processes rather than structures that they lead to. So that's the answer.
Next up, thanks for your super chat from our dearest friend. Everyone in the chat's calling me a beta. Okay, so Kevin Gilfu, stoked to see you again. Kevin says, really great debate tonight. Thanks to both debaters for coming on. James, I want to get yoked like you. Share your workout routine. That's very funny. I actually have to tell you, Nathan Thompson, nobody is more jacked than Nathan Thompson. He has a tremendous physique. He sends me pictures of it. For real. Okay, so with that, I don't know why, but he keeps doing it. So we hope you're well, Nathan. I saw you in the live chat if you're still here. But I want to say, folks, I have gotten to know Dr. Dan, Sal, and Erika. And I have to say they are really pleasant people, folks. So their links, if you want to get to kind of know them more, hear more of their stuff, they're in the description. And so you can click on those links, subscribe, get to have conversations with them, build relationships. Highly encourage you to check out those. As like I said, getting to talk to them and hang out with them. They are just very patient, flexible, and fun people. So really want to encourage you to get to know them. So thanks so much, Dr. Dan, Sal, and Erika, just for being here, hanging out with us tonight.
Oh, yeah. Well, huge thanks to James for making this channel for us to come upon and also speak.
That's nice.
Just small point of order. Do we have like 2 minutes to do closings? Because we move those to after.
We can definitely do those
just
right
like
now.
I mean, honestly, mine's like a minute.
We've run out of questions, so now is the perfect time.
Great. Sal, do you want to go or should I start? You're muted. You're good.
Sorry, you go ahead first. And
all right, cool. All right. So I'm going to set myself a timer. We're going to try for a minute here. We're going to see if I can do this. All right. So stopwatch.
Dan, mine's about 5 minutes or 4 minutes. So
I'll be nice and quick here. So basically, recapping the evening, we actually agreed on a bunch of things, including the questions that I really wanted to address, which were looking at Sanford's conditions that he laid out originally in his book on genetic entropy. And we agreed that if you use the evolutionary definition of fitness, then that process doesn't work the way Sanford describes. Then we got into whole stuff about how much the genome is functional and what is the appropriate definition of fitness. I think the conclusion here is that the redefinition of fitness away from reproductive success does not actually solve the problem, because at the end of the day, we all agree that the problem that genetic entropy proposes, regardless of the mechanism, the ultimate problem is extinction. And if we're concerned about extinction, then it's ultimately all about reproductive output. So by the end, we actually ended up at a completely different argument. That evolution is reductive, that evolution can't build irreducible structures. And all that stuff is debatable, but that's not this debate. This debate was all about Sanford's idea of genetic entropy. And because the conditions that he lays out as required for that process to occur, because those conditions are just not true, they're just not accurate, then the idea of genetic entropy, that there is inevitable genomic degeneration happening, particularly in humans, that idea is without merit. Thank you.
Okay, thank you very much, Dan. Let's pass it on over to Sal.
Okay, my closing is going to be a little longer. Sincere thanks to everyone who made this evening possible, and congratulations to Erika on her engagement. The author of the genetic entropy hypothesis, John Sanford, was once an atheist, then turned Christian at age 39, and then creationist at age 49. Like me, he realized there's no salvation in the name of Charles Darwin and evolutionary theory. With this in mind, I'll close by reading the words John Sanford wrote to close his book, Genetic Entropy. He wrote, quote, "When I was young, I accepted the fact that I was going to die and that all of the people I loved were going to die. I accepted it, but it robbed me of joy, to say the least. I was taught that there was still one hope, that the world was getting better. Science was advancing, culture was advancing, even mankind was getting better. Through our efforts, we could make the world a better place. Through evolution, we could evolve into something better. Through progress, we might eventually defeat death itself. Perhaps we might someday even reverse the degeneration of the universe. My personal hope was that I might, in some small way, contribute to such progress. I believe that this basic hope was shared to a large extent by my entire generation." One of my reviewers told me that the message of the book is both terrifying and depressing. He suggested that perhaps I'm a little like a sadistic steward on board the Titanic, gleefully spreading the news that the ship is sinking, but that is not correct. I hate the consequences of entropy, degeneration. I hate to see it in my own body, in the failing health of loved ones, or in the deformity of a newborn baby. I find it all absolutely ghastly, but also absolutely undeniable. Surely a real steward on the Titanic would have a responsibility to let people know that the ship was sinking, even if some people might hate him for it. I feel I am in that position. Responsible people should be grateful to know the bad news so they can constructively respond to it. If we had been putting all our hope in a sinking ship, would it not be expedient to recognize this and abandon the false hope? It is only in this light we can appreciate bad news. Only in the light of bad news can we really appreciate the good news, that there is a lifeboat. Even as we cannot create life, we cannot defeat death. Yet I assert there is one who did create life and who designed the genome. I do not know how he did it, but somehow he surely made the hardware, and he surely must have written the original software. He is called the author of life. I believe the author of life has the power to defeat death and degeneration. I believe this is the good news. It is my personal belief that Jesus is our hope. I believe that apart from him, there is no hope. He gave us life in the first place, so he can give us new life today. He made heaven and earth in the first place, so he can make a new heaven and a new earth in the future. Because he rose from the dead, we can be raised from death, even the death which is already enveloping us. In these profound yet simple truths, I believe there is a true hope. I believe this hope is unshakable because I believe it is founded on the one who is eternal. It is a hope that has withstood the attacks of time and the corruption of religion. It is a hope freely available to anyone who would receive it today. I humbly put before you this alternative paradigm for your consideration. Jesus is our one true hope." Unquote. And with those words by Dr. Sanford, I conclude my presentation today. Thank you.
Thank you both. And we had a few last minute super chats come in. We're going to quick read and then we will rock and roll. So want to say thanks for your super chat. Nathan Foster said, "Dr. Dan, please look up the definition of quote unquote theory. In that you will come across the word supposition. Look that up and read it out loud. I dare you."
Do that. Thank you.
Next, Stupid Horror Energy, thanks for your super chat. Said, "Split between the moderators. Good job, UWU." Is that some sort of slang
James,
for...
James, I believe that is pronounced ooh woo.
You serious?
Yes.
We are so thankful to have.
Deadly serious.
We're so glad to have Erika, who is a Zoomer. That's the right word, right?
I really straddle that line between millennial and Zoomer, so I don't know who I am.
Oh, that's right. I, however, am a Boomer, which I am quite proud of. So the joke's on you. Next, I want to say thanks for your. Do we get? Okay, we did. We got all of them. So I want to say thanks so much. There was one last thing I thought. Hold on one sec. Oh, yeah. I
believe Stupid War Energy had one more.
Oh, you're right. That is it.
Epistasis.
Thank you so much. Okay. She was gonna crush me if I didn't read this. Let's see, where is it? She says, my understanding of epistasis is that as beneficial mutations accumulate, they tend to be less useful than by themselves. It's not that they become bad or some kind of barrier to evolution, Sal.
I'm not familiar with the idea of, I've heard epistasis in terms of genes having influence on another. I've never heard it used in that context. So my suggestion to Stupid Whore Energy, maybe you could ask questions of Dan, because you'll probably get your money's worth. I'm not kind of delivering for you, but I really want to thank you for your questions and for supporting this channel. People like you have helped make this debate happen. So, but on the other hand, if you're trying to make me look stupid and I couldn't answer your question, maybe that was worth your money. So, but thank you anyway for your question.
We had one last one come in. Corporal Ann, thanks for your generous super chat. Really appreciate the support. It said, how did you say the UWU, Erika?
Yeah, I believe what Corporal Anon is going for is ooh woo, I love you all.
Thank you very much for our translator. And we are excited with that. I want to say thanks so much for hanging out with us, folks. We really do appreciate it. It's always fun. So we will be back tomorrow night as Converse Contender will be here. And I will be here to, as I said, make a quick announcement before he moderates that debate. So thanks so much, everybody. And we hope you keep sifting out the reasonable from the unreasonable. One last thanks to our speakers, though, Dr. Dan, Sal, and Erika.
Peace out, guys.
Thank you for having us. Thank you, Sal. And shout out to Science Friends for your help.
Excellent.
Thank you.
Thank you, guys. And with that, keep sifting out the reasonable from the unreasonable, folks. Take care and have a great night.